beautifully hosted here at the Biostatistics and Health Informatics at King's College London by myself and Professor Daniel Stoll. The title of today's talk is Effective Prediction of Chromatin Interactions in Unseen Cell Types Using Deep Learning. That seems to be very interesting and a little bit different of the topics that we normally include in this series of seminars, right? This is more about basic science, but it's a nice intersection on how to use AI on these particular problems. And we are really, really excited to learn more about this, and particularly from a very expert professor as Dr. Valentina Boeva, who is a tenure track assistant professor at the Department of Computer Science at the ETH Zurich, where she's leading the computational cancer genomics group. Valentina's research is focusing on developing computational methods for multi-omics data integration, something that timely, trying to understand the epigenetic and transcriptional plasticity of cancer cells. We are really, really eager to listen to your presentation, Valentina, so please make a start wherever you like. Thanks so much.
Thank you, Raquel, for a kind introduction and for inviting me to present my work. Let me see where, I can change slides. Also, I think I can see you. If some of you can turn your cameras on, I will not feel like I'm talking to the black screen, which would be nice. And then I will have more like giving a seminar versus rehearsing a talk at home. Thank you, guys. So my name is Valentina Boeva, and I'm working on cancer research. We develop methods to analyze cancer data and to get fundamental insights into cancer biology. And what we try to understand is what makes a cancerous cell cancerous. What we know is that cancer cell can somehow hijack or steal mechanisms of healthy cells to invade, like fibroblasts, to proliferate, like epithelial cells, to resist death, like neurons, or to become immortal, like a stem cell. And how does the cancer cell do it? It does it through mutations. So as far as I know, every cancer cell has mutations. And this can be these point mutations, or it can be larger rearrangements of the genome. It can be insertions, deletions, translocations between different chromosomes, and so on. But all these DNA changes, they result in RNA changes and changes of the behavior of the cell. So this is what we try to understand, is how these mutations translate into changes of the phenotype of the cell. And we already know that about 2% of mutations fall in the regions which are called protein coding regions. These are regions which will code for RNA, which will code for amino acids, and then the amino acid chain will fold into a protein, and this protein will have some function. So 2% of mutations falling in these protein coding regions of the DNA, they will affect the protein function by changing amino acids. And we can predict the effect of these protein coding mutations using such methods as AlphaFold or ESM-fold and so on. But then we have 98% of mutations which fall in these non-coding regions of the genome. And generally, we don't know what they exactly do. And this is one of the challenges that my lab tries to address. We try to understand how these mutations which fall outside of the protein coding regions, how they can change the expression of the genes and then affect the phenotype of cancer cells. And ultimately, what we want to have, not just fundamental discoveries, but also maybe we can find some biomarkers based on these non-coding variants, which could be used for patient stratification and choice of the treatment. So how can we know what these non-coding mutations do? We need to understand what it does to the accessibility of the DNA and also to the DNA folding. So first, accessibility. Accessibility of the DNA means that the DNA is accessible for binding of different regulatory proteins. Usually, DNA is not accessible, so it's like close, close, close. And then there is one region which is called promoter, if it's in the beginning of a gene, or enhancer, if it's far away from the beginning of the gene, where many different regulatory proteins bind, where some transcription can happen. And so we call these regions accessible, and there is an assay called TAXIC, which can allow us to profile the accessibility of these regions. So what mutation can do? It can disrupt binding of some regulatory proteins, disrupt this beautiful hub where many things happen on DNA, and then this is not going to be an active regulatory element, not an active enhancer or promoter. So this is one way how a mutation can make consequences on gene expression, for example, mutating and disrupting a promoter of a gene where transcription starts. And another way how a mutation can affect expression of genes is by disrupting the folding of the DNA. So we have promoters, which are located in the beginning of the gene. We have enhancers, which are located further away from the gene. In 3D, they interact, they are close to one another. And a mutation can disrupt this interaction, and then the expression from the promoter from the beginning of the gene will decrease. And we try to understand what mutations can disrupt regulatory elements and which mutations can disrupt this 3D folding of the DNA. And today I will focus on the second question, disruption of the folding of the DNA. And when I say DNA, we can also say chromatin. So chromatin is just DNA with the nucleosomes and other proteins sitting on it. This work was done by my team, Ayush Gruber, in collaboration with the Swiss Data Science Center, Lin Zhang. So before I go to the method, I would like maybe to mention more about the biology, which we need to know to understand how we build this method. So DNA is a long molecule, as you probably know. And I already mentioned that along the DNA, we have these hubs where something happens, where regulatory proteins bind to the DNA. So we have these enhancers, which are away from the beginning of the genes. We have promoters, which are close to the beginning of the gene. And then we have these different regulatory proteins which bind there. The assay, which allows us to profile where enhancers and promoters are located, is called ataxic. And these regions, we call them DNA accessibility regions. So when we do this experimental assay, which is relatively cheap to do, we get this red profile, which shows us what is open or accessible on the DNA. So usually the DNA is closed, so non-accessible, and then we have these peaks of accessibility. It means that this is an important region. All important regions, they have these peaks, means something is going on DNA. And this is in 1D. In 3D, these regions interact. And if you have a beginning of the gene here, where I show this purple protein called RNA polymerase II, which transcribes DNA into RNA. So if we have a lot of interactions of these promote regions with many enhancers, then we have a lot of RNA created from this promoter. So if you have just one enhancer, we'll have a certain amount of RNA. If you have 10 enhancers folding to this region, then you will have a lot of RNA being produced. So we have information from this, we have a flow of the information from the folding of the DNA to the transcription levels. And we know that certain factors can determine this folding of the DNA, and among them we have CTCF and YY1. They're experimentally known to be located at these loops that DNA makes. And this is important for our method development. So what we want to have, we want to now predict the strength of the looping or interactions between different accessible regions of the genome, meaning promoters, this is where the transcription starts, this is the beginning of the gene, and enhancers, which could be, which are regulatory elements which can in 3D fold with the promoter and enhance the transcription. And we operate at very large scales, so the distance between a promoter and an enhancer which folds this promoter can be up to 2 million nucleotides. And we don't know in advance what is the strength of interactions between these regions, and this is what we want to predict. If we know it, then we can explain the effects of non-coding variants of falling in these enhancers or promoters on the genes. For example, if you know that this enhancer on the left, I hope you see my mouse, is folding in 3D with this promoter, then we can say if this enhancer is mutated, then this gene will be affected and not some other gene. So we need this information. And to profile this information experimentally, we can use such assays as HiSeq. HiSeq costs more than 10,000 francs or euros, dollars, or pounds per experiment. And this is why HiSeq experimental data for this DNA folding is not available to all cell types and cell states we want to study. So we want to make predictions. We want to know how a mutation in the DNA will change the folding of the DNA in 3D. And these type of predictions, they are similar in some sense to predicting folding of the proteins. So there are some similarities and there are some dissimilarities. And I will first focus on the similarities. So you know about protein folding most likely because there's AlphaFold method and Nobel Prize. So when we try to fold protein using machine learning method, we always start with a 1D sequence of amino acids. So we have 300, 400 amino acid sequence on average, and then we may predict the contact map. So the contact map tells us the strength of the interaction between two amino acids in the protein. So you see the strong diagonal here, meaning that if two amino acids are close to one another in 1D, then they also close now in 3D. But then you have alpha helices, you have beta sheets. It tells us about 3D interactions of residuals which are quite far away from one another. And then from this contact map, we can predict already the 3D folding of the protein, or such measures as AlphaFold can go directly from the 1D sequence of amino acids to the 3D coordinates of the residues. This is for protein folding. For DNA folding, it's somewhat similar. We also have as input this sequence of nucleotides, usually 1 million nucleotides or more. And we want to predict, again, the contact map. So the contact map tells us which nucleotides are interacting with one another in 3D. And this contact map can be obtained experimentally using the high-C technique, and we use it for training. We cannot predict the true 3D coordinates, unfortunately, because we cannot crystallize DNA, so we don't have the data available to train the true 3D coordinates for nucleotides, but we predict these contact maps. So these are the similarities. Talking about these similarities, first, when we fold DNA, we use much larger receptive field. It's more than 1 million nucleotides that we try to fold, while for amino acids, it's about 300, 400 amino acids. Then also for the proteins, the predictions do not depend on the cell type. The same protein will fold the same in a muscle cell or neural cell or skin cell, while DNA doesn't fold the same in different cell types of our body. So in a neural cell, the DNA can fold differently from an immune cell. And this is the complexity. Also because these experiments, which can provide us the contact maps to train our model, are very expensive, and they are available maybe for 10 different cell types, but not for 200 cell types that we have in our body. While for proteins, it doesn't depend on the cell type, so we just have this data available to us. So it looks like a very complicated problem, right? So we have much larger receptive field, we have cell-specific binding, training data kind of scarce. So can we do it? The answer is yes. And there was already a paper published in 2023, not by us, but by a team from NYU. And this team showed that if you just throw a transformer architecture, so out-of-the-shelf transformer architecture on the data, then we can predict quite accurately the DNA folding. But for this, we need 3 different types of data as input. We need the DNA sequence, so the sequence of nucleotides, A, C, G, C. And then we have also to provide the accessibility profile, this red peaks, which tell us which enhancers and promoters are accessible, and the shape of the accessibility profile. And on the top of this, they require experimental binding profile for this transcription factor CTCF. This is the protein which determines the looping. We already know from biology that this protein is very important to create loops on DNA. And so we can train this model, which is called Origami, and then make predictions. Unfortunately, because the model is very bulky, it has more than 13 million parameters, then one prediction for one cell type will take more than 60 hours on GPU. Also, the model will not predict uncertainty because the authors didn't maybe plan to use it to predict differential effects of mutations on folding. So the model is nice, but not ideal, and we try to improve it using our biological understanding of what drives folding of the DNA. And I'm just going to show you the preview of our solution, which we call the UniversalEPI. First, in terms of the accuracy, so this is the Spearman's correlation on the y-axis. Our method is green. This other method, which was published in Nature Biotechnology, is in red. This is the same method, but when we retrain it without using this modality of CTCF, and this is another method which uses DNA only as input. So already what you can see is that our predictions are more accurate, significantly more accurate than the predictions of these other methods. And also this like Spearman correlation of 0.95 means that we are truly accurate. So, and this is most importantly predictions for unseen cell types. So for example, we train on immune cells and we predict in skin cells. Also, our method requires fewer inputs. So if for C origami, we need this additional input for the regulator protein CTCF, here we don't use it. We just use the DNA and the accessibility. DNA, we need it because we need to predict the effect of mutations. The accessibility, we needed to make a cell type specific predictions. Also, our method outputs the uncertainty estimations, which are very nicely calibrated to mean squared error. So when we predict high uncertainty, we also know that the model makes more mistakes. And also we are much, much faster. So here I'm showing to you as a top row the runtime in minutes. For C origami, for just to do the inference, it takes about 60 hours, and we can do it in 8 minutes if we use just one model. And if we use a deep ensemble of 10 models, then we are just 43 times faster than this other model. So I think when we saw it, we thought, okay, it's a cool model that the community should use, but how can we achieve it? How can we build such a model given that just a standard transformer out of the shelf model is so slow, so bulky, and makes worse predictions than we do? So now I'm going to show you how we used our biological understanding of the mechanisms of DNA folding to build a machine learning model which will outperform out-of-the-shelf transformer model. So the first idea that we used was that we should ignore all regions of the DNA which are not accessible. So everything which is in between promoters and enhancers, we can just forget about it and use only the information between, about the distance between these two regions, right? This is one idea that we used. And then the predictions that we make, this will be the pairwise interactions between these accessible regions. So we are not going to predict the full contact map, but just the contacts between these accessible regions. And this makes our model very, very lightweight. We can increase the receptive field up to 3 million nucleotides, and we will have something like 3 million parameters, or maybe even less. And this distance between the peaks, we just encode it using positional encoding in transformers in a classic way. So now we have a larger receptive field with a fewer model parameters. And then we had another idea which allowed us to train a model, again, with fewer parameters, but which doesn't overfit to the cell types used during training. So we train, for example, on blood cell types, and they validate on liver cell types, and we are still very accurate in our predictions. And to do it, we decided to use only the information from the DNA, which corresponds to binding sites of 3 regulatory proteins, which are known to be expressed in every cell type, even cancer cell type. And these 3 proteins are involved in looping of the DNA. So I already mentioned CTCF and YY1. They are known to be involved in looping, and SP1, it's a promoter marker. So it helps the model to know which region is enhancer and which region is a promoter. So we have a 2-stage model that we train. The first neural network that we train simply predicts where these 3 transcription factors, the 3 proteins, bind on the DNA, given as input the DNA sequence and the accessibility profile. And we do it through training a 5-layer convolutional neural network without any skip connections, that we just get information from the DNA and we make predictions. And this allows us to learn in these convolutional layers the information about DNA features, which are important for binding of these three proteins, and ignore other DNA features. And then the second step, we pass information from these 5 convolutional blocks to our transformer model through the gates. So we have the gates. Through the gates, this information is passed to the next model. Here we have 5 blocks of transformers, and then we make the predictions between different accessible regions of the DNA. Why we need the gates? Because we don't want all information to be passed to our transformer model, because we don't think that every layer has information which is important to make predictions. And we don't know in advance which layers capture which information. Also, we don't know how this is linked to different transcription factors we are interested in. So the idea was to use these gates, and I'm going to show you how we implemented it. Here we used a stochastic gating idea, which was published at ICML in 2020. So what we do here, we introduce 5 variables for 5 convolutional layers that we have, which takes values between 0 and 1. And this is the distribution that one can have. The distribution is parameterized by nu k. So we have this k going from 1 to 5. And if nu k is high, we have a lot of values of 1. If nu k is low, we have a lot of values of 0. And overall, you can see this distribution as a continuous relaxation of the Bernoulli distribution. So we have these gates. If the gate takes value of 0, then the information from this layer is not passed to the transformer. And if the gate value is equal to 1, then the information is passed to the transformer. And so, and thus, these gates, they contribute to the final reconstruction loss. So they show us which information from which layers will be used to perform the reconstruction of the signal of interest, the pairwise interactions between different regions of the DNA. And on the top of this, we have this regularization to make, to close the gates that we don't need. So this regularization just adds a value scaled by lambda, and if nu k is high, then the penalty will be high, and if nu k is low, so we have a lot of values of equal to 0, then the gate, the penalty will be low. So during the training, we always sample from this distribution parameterized by nu k to get zk at each step of the iteration, and during the inference, we just fix zk equal to nu k. So this allows us to learn which layers we should use to make predictions in our transformer head. And then another idea that we had to use is how to estimate the uncertainty, because our final goal was to say how the folding of the DNA changes when we have a mutation. And if our model just predicts that with mutation we have interaction strength of 1, and without mutation we have interaction strength of 1.1, we couldn't really say whether this is just the noise of the model or the interaction truly changes statistically significantly, if you wish. And for this, we needed to make our model to output the uncertainty prediction on the top of just making the prediction itself. So we want not just mu, but we also want this sigma squared
I don't know, Valentina, if you can see me or even hear me, because we can't hear you. We can see you, but we can't. At least I can't hear you. The
Okay,
rest can
so
hear. Now we can hear you, isn't it?
yes,
It's all right now, Johan. Yeah, perfect.
sorry guys,
Thank
it's
you.
just somehow
It's
I
all
saw
right.
that the
I
connection
think we missed
disappeared.
like 1 minute, maybe.
All right, yeah. I will try to reshare my screen. Hopefully this will not happen again. Somehow Teams is not cooperating today.
Sorry, don't worry.
Yeah, but it's not your fault, it's Microsoft fault. I need to see now how to share the screen. You don't see my screen, right? No, okay, now I see how to do it.
No, we can see it now.
Yeah, it's coming. It's coming. Okay.
Okay, thank you.
Right. Is it good now?
Yes, it is.
Mm-hmm.
Uncertainty estimation, biological relevance, right?
Right. Yes.
Okay.
Super. So what I was saying is that because we were interested in differential effects of mutations to compare what happens to DNA folding when mutation arises, we needed this uncertainty estimate. So in addition to prediction value mu, we needed to output the uncertainty sigma squared. And we model this uncertainty as a sum of aleatoric and epistemic uncertainties. And I think to this audience, I don't need to remind it, but aleatoric uncertainty measures the variability in the data, while the epistemic uncertainty corresponds to the lack of knowledge of the model where we don't have enough data for training. So we need to model both of them. And how we do it? This is the total uncertainty that we output. The epistemic part of it corresponds to the variance, which is estimated using deep ensembles. So here, instead of using just one model, we train 10 different models with 10 different random seeds. And then we take the predictions of each of the models, and then we just estimate the variance using these 10 different predictions. So this will be our epistemic uncertainty. And then we also output the aleatoric uncertainty, which we get from changing our loss from mean squared error loss to the beta negative log likelihood loss of the normal distribution. So when we have this loss, which looks like this, we don't only model mu, but we also model sigma for each prediction. And then we can just take these sigmas, which are like mu and sigma are used in this non-negative, sorry. So the sigmas and mu's are using negative log likelihood loss through this equation here. And then we just take the sigmas and we calculate the mean values of these sigma squares, and this will correspond to our aleatoric uncertainty. So we have these two uncertainties, and I already mentioned that the sum of them scales very nicely with the ground truth uncertainty of your model, which we can measure with the mean squared error between the predictions and ground truth values. So this is how the model works, and the accuracy, I already showed it to you, just to show you some examples on 4 different cell types, what this accuracy of Spearman's rho of 0.9495 means. So it means that we can really accurately make predictions for the DNA folding between different regulatory elements. And since we can do it, then we can now perform in silico experiments. We can take the DNA, then take a region here. This region is responsible for a big loop of the DNA, which is called TAD, for topologically associated domain. We can just inverse the DNA and then see what will be the effect of this in silico inversion. And what we see is that after the inversion, we have predicted decrease in the interactions on the left of these inverted regions, and then increase in the interactions on the right. So we change the folding of the DNA according to the predictions. We can also look at the confident predictions using our certainty estimates, and then these links here depicted in pink and green will be statistically significant changes in the folding that we can output to the biologists or bioinformaticians who want to use our model. So now what I showed to you is the model which allows us to model DNA folding from DNA sequence plus DNA accessibility. These are these two inputs that our model requires. We think that now this problem is solved because we can make very accurate predictions, and also the model is very lightweight. You can make predictions in 8 minutes for a new cell type. But this is not the end. If you want to understand the effect of mutations on RNA, because first, DNA mutations can change accessibility, which is also an input to our model. And we need to know how we can predict these changes on DNA accessibilities from the mutations. And for this, we build another model. This is, you can find it in the preprint already available. And this model uses, actually we have different models that use different architectures, but the main block of it is this conv-next v2 block, which includes some skip connections, convolutional layers, and different layer normalization here, for example, global response normalization. And this allows to much better capture the information from the DNA to make the predictions of DNA accessibility. What we noticed is that, so we can improve the architecture of almost any model like LSTM or dilated CNN or just a classic CNN by including this layer, our convex v2 convolutional layer here. So then the prediction becomes very good, independent from the downstream type of the model. So here you have transformer-based model. Here you have a model which uses dilated CNNs, and this is just a CNN model. And you see that the performance plateaus if we just include this new block here in the beginning of each model. And what is cool about it is that now if we predict changes in DNA accessibility, and then from this we predict changes to the DNA folding, the error doesn't propagate. So what I'm showing to you here, this is the accuracy of UniversalEPI using ground truth accessibility values. So we predict folding and we measure the accuracy of the predictions for the folding. In purple, this is based on experimental accessibility track, and these four bar plots show you the accuracy of the predictions for DNA folding using the predicted accessibility. So we take DNA, we predict accessibility, and then we use these predictions to predict folding. And you see, for example, that for transformer models, we almost don't use any accuracy when we make predictions of folding. So this means that we can already quite accurately go from DNA to DNA folding, and the only remaining part is how to go from all this to RNA changes. And we also have a model for that, which is not publicly available because we still work on it, but we have a model which takes into account just DNA and accessibility of the DNA input, and we perform on par with another state-of-the-art model which uses an additional epigenetic modality as an input. So hopefully one day we'll have a comprehensive model which could allow us to model the effects of all mutations on all transcripts in all cells. The immediate impact of this would be to replace very expensive experiments. For example, these contact maps are very expensive to obtain, or also if you want to see the effects of mutations on the expression of the RNA, CRISPR experiments are also very expensive to do. So the immediate effect is save money, perform screen thousands of mutations in silico, understanding maybe for the fundamental effects on cancer, then building this model from DNA to transcriptome, and eventually we hope that with this we could profile the effects of all mutations which are non-coding mutations in our genome, how they can affect gene expression in cancers, contributing to understanding better the fundamental mechanisms of cancer development, and also maybe getting some non-coding biomarkers for patient stratification. So, I just wanted to thank people who worked with me on this project. This is Ayush, who was the leading person in my group, then Lynn from Swiss Data Science Center, And these are all our collaborators who contributed different parts to this project. And on this, I would like to thank you for your attention, and I will be happy to take your questions.
Thank you very much, Valentina. So yes, if there's any question from the public, please, you can just open your microphone and formulate. Yes, Mariam, please, you can go with your question. Thank you.
That was excellent. Thank you. Where have you actually? How are the models employable? As in, are they written as software or packages within, say, Python or R, etc.?
This is all on Python, and if you find this publication on bioRxiv, they will be linked to the GitHub, and on the GitHub will be different instructions how you can employ the model with all weights already available, or how you can retrain the model if you need to retrain it.
Okay, that's fantastic. We have a lot of mutations that we'd love to test with this. So it'd be great. And perhaps we could actually send you a list of mutations for your third model that's as of yet unpublished. That would be really good to do.
Yeah, please contact us. So maybe just to make it clear, we have two models which are now publicly available. So one is predicting changes in accessibility of the DNA from the DNA sequence, and then this model which predicts changes in the folding given accessibility and DNA. These are two different models.
Yeah, with this last model, is it something that you could actually do with cell-specific expression?
This is all for cell-specific, yes.
The RNA expression, is it for cell-specific or whole blood, for example, or everything together?
So we always do at cell type specific. We don't predict it for mixtures. At least the model design in such a way and train on cell lines, which are supposed to be homogeneous, so that if you tell us what is the accessibility of your cell type, which you can take just online, right? And then what is the DNA sequence that we can predict the folding and eventually the expression.
Excellent, thank you.
Thank you, Mariam, for question.
Is there any other question from the public? Any comment? If not, I might have a comment about the methodology that you use, Valentina. I found it extremely challenging and very interesting the way that you, as you use deep learning here. I was just thinking, so it really fitted very well your case, even if we didn't know that there is a black box problem. Would you think that the fact that this is a black box AI model was posing you any difficulty? The fact that you cannot understand, or how do you try to understand the final outcome, the fact of being a black box, or even if you have tried any other machine learning model as an alternative? But does it make any sense in your example?
Right. So I'm not sure. So it, in some sense, because we build the model using our biological intuition, and it has this structure of these convolutional layers and then gates.
Exactly. That's the slide.
It's not that black box because we can check what actually gets activated. We could, for example, we see that there is a method where you change something in your input and you see what is the effect of the output. We can also see what will change in the activation maps of the transformer or in values of the convolutional blocks here. And we see that, for example, the model captures CTCF very nicely. And interactions between two CTCF sites, we can see it. So
yes, I could see it in that slide when you showed the reconstruction and how you were dealing with this. And I thought, well, we are always talking about the black box of the deep learning, but it depends how you fit this to your problem. It can become a not black box method. This is what I really like it of your approach.
Thanks.
Thank you. I think there is another. Yes, again, please ask, Mariam.
Yeah, sorry, me again. I did also want to inquire about the regularization. If you just explain a little bit more about how you did that, that would be great.
So for this one, right?
Yes, that's it, yeah.
Right, so just maybe to reiterate how these gates function. So we try to learn the 5 values, nu1 through nu5, which will open each of the 5 convolutional layers so that information can flow. At every time during training, we sample a value using this distribution from this distribution, which is parameterized with nu k. And actually what it is a truncated normal distribution. So we sample from a normal distribution with mu equal to nu k and sigma equal to 0.5, and we just truncate it. If it's below 0, we say 0. If it's above 1, it's 1. And when we do the inference, we just use the value of nu k. And so this regularization here is just the probability that the gate is open. And because we first sample from the normal and we truncate it, then the probability that the gate is open will be simply the cumulative distribution, the cumulative distribution function of the normal distribution with the mu equal to nu k and the variance of 0.5, the sigma which we used in the beginning. So it will penalize us for using very high nu, opening the gates. I'm not sure whether this was clear.
Yeah, that's great. Thank you.
Thanks.
Thank you.
Any more questions?
There is someone asking for the link to the bioarchive paper. If you have it readily available, that would be good.
Right. So I think what you can do to find it is just to type bioarchive, Voeva, and then the name of our method, which is called UniversalEPI. I will just find a slide where the name appears. Here, this name in green, UniversalEPI. So by just typing UniversalEPI bioarchive, one should be able to find the paper.
Excellent. Thank you so much, Johan, for sharing so you can find it in the chat. Is there any other question from the audience? Yeah. Yeah, Vera Milovanovic, please.
Yes, hi.
Yes,
Hi, Valentina. Thank you for your talk. I just have one question. So do you have any ablations on how important was to take like lots of nucleotides and like these, like also expressions? Like you mentioned, like you can go up to 3 million, I would say. And like, yeah, was it something that was very important? What would happen if it was bigger or lower, or is there any like technical issue with going bigger? Like, would it even help, let's say?
Mm-hmm. Right. So for this specific question, how large the receptive field should be, we didn't make ablations. We just make it as large as we can so that the model is easily trainable. So I don't have the answer to your question whether we can go larger. What we see is that the accuracy for the predictions between very distant regions declines slightly, but it still stays very good. So we don't see like a drop in the accuracy given the distance if the distance is very large. We did other ablations. For example, we tried to remove some proteins that we use to train our network. So these 3 different proteins, we tried to remove some of them or put some other important proteins, and we saw that the 3 are needed and other proteins are not needed. We did some other ablations. What if we don't use deep ensembles? It's worse. What if we use MSC loss instead of negative log likelihood loss? It's worse, and so on. And I think if you look for supplementary figures, you will see a lot of ablations discussed there.
Okay, thank you very much.
Thank you very much for the question.
Thank you for the questions and for your answers, Valentina. Is there anyone else? I need to ask anything, any comment? Is there anything in the chat? Let me check. Sorry, because my mobile is not that flexible. Okay,
Yeah.
there's nothing there. So
maybe I can.
Yes, there is one question.
Yes. Sorry. Yeah, hello. Thank you
Yes,
for a
Johan,
great
please.
talk. Yeah. So I was just wanting to ask: is there any way to get a sense of the sort of representability of the training data? Because you said that the availability of this experimental data was not very good. And do you know, I mean, because you received fairly nice performance statistics of this method, but I mean, if this training data is not representable in the sort of, if you could get all of the data in the world at some point, I mean, is there any way to get a sense of how representative the data you have as of now is sort of valid?
So I think the answer to your question is we now here we report the accuracy on just 4 cell lines. So first is the cell lines. These are not like natural cells which are sorted. You have cancer cell lines, like this one is a cancer cell line. This is a healthy cell line. And this is, for example, the liver cells, and this is the blood cells. So the fact that we train on very different cell types and we can predict on like liver coming from blood means that already we learn the major mechanisms. I guess the reviewers, if we go to the reviews, because we're still with the editor, when reviewers will ask us for these questions, then we're already looking for other available high-C data sets on which we can validate. We found beautiful data sets from Barcelona on mantle cell lymphoma and CLL. Some other data sets where we try to validate, show that the accuracy is very good. In our experiment, we have, in the paper, we have an experiment of macrophages, which are not a cell line, but like primary culture. The accuracy is lower. It's like 0.92, or not 95, but it still works also on this not a cell line type problem. And also this experiment that I just mentioned about macrophages, they also differentiated or activated using some chemical. And then we also predict what will happen to them in terms of the folding. So even if we perturb the system, we can still make accurate predictions. And I thought this is really cool. But you're right, the more, it would be nice to have more data to show more different cell types where we can make accurate predictions.
Yeah, but it seems like the sort of things you mentioned, it means that your model is fairly robust also in these sort of different cases.
This is what I hope it is. Yeah, exactly.
Yeah. Okay, yeah, thank you so much.
Thank you, Johan, for the comment. Thanks, Valentina. Is there any other comment or question?
Yes. Can I ask a question?
Yes, Shafia, please go ahead.
Yes.
Yes,
Thank you. Thanks.
Perfect.
Yeah, thanks a lot for your presentation. Regarding your GitHub tool, I just checked your tool, and I would like to know if we can give some genetic loci, and then ask the program to give us which genes are interacted with our loci of interest.
I guess if you have DNA accessibility for your cell type?
Well, basically, we don't have something like ChIP-seq data. We basically have some SNPs, some regions that are hypervariable and are related to our phenotype. And I would like to know if there is any way that we can find that which genes are interacting with this specific locus.
So this model is exactly for that. The only thing that we need to know is what is the cell type? What is your cell type?
For example, it could be a neuron.
Right. So if you can find DNA accessibility for your neurons in ENCODE or any other place and provide it, then we can make predictions. If you don't have accessibility, so this ataxic or DNase profile, then we couldn't make predictions because we wouldn't know anything about cell specificity, because you want to make cell type specific predictions, right? Because your locus of the SNP can fold with one gene in one situation and with another gene in another situation. And this is cell type specific. So if you don't care about cell type specificity, but you care about just something which is general, like on average, right, in every cell type, tell me, then there is a model which is called Akita. So I mentioned that in the very beginning when I was doing the benchmarking. So this yellow one, you see, Akita.
Yes.
It runs only on the DNA. So in this case, you can just run the Akita model on your DNA, and it will give you some average predictions, which will not be cell type specific. If you want cell type specific predictions, then you need to find somewhere the accessibility profile.
Yes. Okay, great. Thanks a lot.
Thank you for your question.
Really practical questions. I think that's been very helpful for the attendance, Valentina. Thank you. And for you as well, because you know that people are going to be using your code.
I hope so, right? Yeah.
Which is perfect. Excellent. Is there anyone else? We still have 6 minutes. Yes, please, go ahead.
Maybe just this figure. So with this origami, I mean, I know probably it's really not your model, but I'm puzzled because when you like remove this CTCF, you're kind of on average the same, but then also maybe like also this error, or I mean, you're more consistent. I mean, what happened with that? I don't know. Is it important to have this additional information, or?
So what we see is that for C origami, it's important to have this third input, the experimental binding of the protein which causes the loops, TCF, because then the median accuracy decreases, right? This is what you see. This is the median.
Okay, I haven't seen. Okay. And okay, it's
the median decreases, right? And also you have some outliers when the predictions are really worse. So this is the different random seeds or different chromosomes. So overall, it gets worse. We didn't try to provide CTCF to our model because we thought it's not needed. We can just, our first neural network can just learn the DNA features which are needed for binding of CTCF, and the gates will filter this information and you will be fine.
Okay, cool. Thank you.
Thanks. Any more questions? Raquel? I think the connection got lost.
My connection. I'm back. Sorry about that. Okay. 4 minutes to finish. If there's any other comment, can you hear me? Sorry, because I have.
Yes, we can hear you.
I don't think so. Okay, perfect. I see you frozen, but okay. Well, if there's no more comments, okay. So let's get it for finish. So Valentina, thanks very much. This has been an excellent talk. As I've said, a little bit different, but I could see that our audience were really appreciating your work, and the discussion was really interesting and useful, I think. I hope that you found it helpful as well.
Yes, thank you, Raquel, for inviting
me.
Thank you very much.
Let's stay in touch. Thanks everyone in the audience as well for this lively discussion. We will be again celebrating another machine learning meeting by the end, the last Wednesday of April. We'll be here again. So thanks everyone and enjoy your evening.
Thank you, bye.
Thank you. See you soon.